Hi there, this is David Nguyen of Tissue Spatial Geometrics Lab, and in this presentation I'm going to give a layperson summary of the paper that we recently published that provides evidence for something called heritable non-genetic information. I'm doing this at the request of students and friends who don't have the background to understand the paper since it was written for an expert audience. Heritable non-genetic information is abbreviated HNI and is pronounced honey because I think it sounds funny. So honey is instructional information that can be inherited. That means it can be passed down from generation to generation. Furthermore, it is information that is not stored in DNA, which is where information in your genes are stored, and it is not stored in epigenetic marking patterns that package DNA. So epigenetics are molecules that are attached to either DNA itself or the proteins that package DNA into chromosomes. And epigenetics are ways of affecting how genes behave without changing the gene itself. And I point this out because honey is not stored in epigenetic markings. And lastly, honey governs how cells and molecules behave in health and disease. So now that I've given the formal definition of honey, I will now provide 5 examples that were covered in the paper. The first example of honey is in growing cancer cells in a 3-dimensional gel instead of in a flat 2-dimensional plastic dish. Now, cancer cells, such as breast cancer cells, can have many mutations, which is why they're cancerous. But when you grow them in a gelatinous matrix consisting of proteins from tissues that are naturally outside of cells, this protein environment is able to convince these cancerous cells to grow into a nice and neat hollow sphere, which is how they're normally supposed to behave. And the cells do this even though they still have the same genetic mutations as when they were grown in a 2-dimensional dish. So this is very powerful evidence that the protein matrix in which the cells sit has instructional information that can override the bad information in mutated DNA. Example number 2 comes from a study done a long time ago in which scientists were studying human melanoma cancer cells. These are very aggressive skin cancer cells that have a lot of mutations and a lot of corrupted information in their genes. And they injected these human cancer cells into what's called the mouse blastocyst. That is, it's a mouse embryo. So here are the stages of development of the mouse embryo starting from one cell. And at this stage, this is called the blastocyst. And when they injected these human cancer cells into the blastocyst, they thought that they would get just a tumor that would develop because the cancer cells have so many mutations in them. But to their surprise, what they got was a healthy baby mouse that grew into a healthy adult mouse. Except that this adult mouse had human cells with mutated instructions in those cells spread throughout its body. So this was amazing because it showed that something very special happens in the embryonic environment, such that it can even subdue very damaged DNA with broken instructions, right? And even cells that have damaged instructions can end up behaving normally. Example number 3 is in the experiments of salamanders and their ability to regenerate their limbs after amputation. The amazing thing about these experiments is that the cells at the location of amputation, or where the cut was made, seem to remember that salamanders have 4 toes. And the amazing thing about this is that this regeneration happens not in the embryo, because we know that something very special happens in the embryonic environment, but this happens just in the aquarium or terrarium in which the salamander is running around. And yet, even without any external instructions, the cells at the wound site seem to remember exactly how to form. Next are experiments about flatworms. Flatworms have this amazing ability to regrow their bodies even if you cut them into pieces. And scientists have shown that when you let flatworms bathe in water that contains a molecule that blocks the electrical signaling between cells, what it does is it blocks these channels that allow cells to share juice with each other. So when you block those channels, you also block the electrical signaling between cells, and that results in 25% of the fragments growing two heads, while 72% of them grow as normal. But the amazing thing is that the cells that are double-headed, if you cut them into pieces, they keep regrowing back as worms with two heads. So somehow, without changing the DNA of these worms, just by blocking the electrical signaling for a period, you create a physiological memory that persists generation after generation. And again, all of this can be done without modifying the DNA in these flatworms. In fact, they share the identical DNA because they're clones of the same parent. And lastly, here's an example of honey at the molecular level. So the information in your genes is carried in DNA. That information is copied into mRNA. That mRNA is then translated into a string of amino acids, which folds into a protein. Now, for a bit more detail, this amino acid sequence will naturally fold itself into either helical structures or these flat beta sheets, they're called. And then these sheets further fold upon themselves to form a complex 3D shape, and multiple proteins can then combine together to work as one unit. And so textbook biology teaches that the primary sequence of a protein, that is the sequence of amino acids in this chain, is what determines how a protein folds. And that's pretty much all the information that a protein needs for it to fold by itself. However, we now know that there are parts of proteins called intrinsically disordered regions. That is, these regions here shown in red, in which the protein does not fold into a three-dimensional shape. We say that they are disordered or shapeless. Some proteins do not have intrinsically disordered regions, but it turns out that 15 to 45% of proteins in eukaryotes, which includes humans, do have these IDRs, or intrinsically disordered regions, and a small percentage of proteins are completely disordered. The amazing thing about intrinsically disordered regions is that they can fold into three-dimensional shapes, like helices and sheets, but they need extra information in order to do so. That information comes from binding to other proteins or from local environments, such as being stuck in an oily membrane. That extra information that comes from the oily membrane or the shape of another protein is what tells these disordered regions to fold into the shapes that can do a function, such as cause a reaction to happen. This means that the full set of instructions for protein folding is not stored in the primary sequence. So this is the primary sequence. If the full set of instructions is not stored in the primary sequence, that means it's also not stored in DNA, because the information for the primary sequence comes from the DNA. I distinctly remember the moment when I had this epiphany while reading about intrinsically disordered regions of proteins, and it was a moment of breathless amazement. It just goes to show how much about biology we still don't know. Because in order for this intrinsically disordered regions to get instructions from an external factor to tell how to fold, some other code had to put the external factor in place such that these two recognized each other to induce a 3D structure in the disordered region. In other words, there is a code layered upon a code that's layered upon another code.